So from where we started yesterday, we'll go ahead. Those who are not able to see presentation, as I told you, please try to click on the presentation. Okay. Now what US guidelines say? We have seen yesterday see, you may able to see I don't know whatsoever you have saved me, or whatsoever it is appearing on your screen from my name. Most probably there are two things are coming. One is the person speaking, and second thing is the person presenting. In both cases you will see two things from my side. So in second one there is a presentation, and in presentation try to click on presentation. There is no, in front of that somebody is presenting, you will find, you click on that, in front of that whatsoever presentation is there, you click on the presentation. Otherwise Akash and Achal, who have tried it successfully, they can inform them telephonically, or on WhatsApp simultaneously they can see. Okay, because already we are too late. When you expect it should be at 5 o'clock, and up to 12 minutes 13 minutes either people are not joining or there are technical glitches, then it's not good. So acute toxicities, the US federal, we have seen so far in earlier class that how acute toxicity, chronic, subchronic are there as per OECD what are various guideline numbers. And very unfortunate that only 3 have tried what I had told. This is really pathetic with this class. It seems that only 3 has got access to internet, or only 3 is using the internet. Others are having really very very big problems, it seems. This is not good. So in acute test toxicity one, we left with this, that is in US, the US federal regulations, they require that an acute test of chemical in question that should be determined at least 2 animal species, with one species being a non-rodent. That means what are the rodents? Mice, rat, they are called as a rodent. So these are the smaller species. So that means that one small species and one bigger species. Okay, like cat, dog, or rabbit, like that it should be used. The results are reported as LD50 to LC50, and it will stand for the lethal dose to lethal concentration, which kills 50% of the animals. So same way, other things are same, only thing is the guideline says that you should use 2 animal species, and out of that one species only should be rodent and other should be non-rodent. Then next in that, how subacute toxicity study has to be done. Chemical is fed to the animal for 3 months at different doses. The dose response curve is developed. Highest dose, which produces no adverse effect, is used to estimate for the safe dose for human consumption, and then other doses above that, they are considered as a toxic doses. Okay. Then subchronic, that is an exposure for 1 year. Other things are similar as we have seen earlier. Chronic toxicity testing, It is done for the lifespan of the animal. Okay, it follows acute and subacute testing. Why acute and subacute it follows? Because we have seen that acute and subacute helps in determining dose, determining the earlier situation. If animals are dying in subacute only, or that dose is causing death, then there is no fun in going for subchronic or chronic. So initially always it is a stepwise process: first acute, then subacute, then subchronic, and then chronic. Again, as I told you, I don't know whether you have referred or not, in Kadzung it is very nicely given that basically what is important is what kind of drug you are having. If the drug is going to be used for lifelong as antihypertensive or anti-diabetic or such kind of drug, then in that cases, or maybe we have seen anticoagulant, etc., then in that cases you must go for lifespan or chronic toxicity testing. If it is going to be used for one time or two times, then up to subacute it is fine. Okay, if it is going to be used for maximum 1 month, 2 months, like antibiotics in some of the infections, then okay, fine to go for the up to subchronic, because it is unnecessarily waste of money, waste of time, because as the time goes, the cost of all activity, all research increases, and that has put in the product. Okay, and this is how the planning is done. So followed the acute and subacute testing. Rodents are normally used because their life is about 4 years lifespan. Means the study will be over early. Testing lasts for 3 generations. This will help you to understand the genetic effect of the drug in the next offsprings. The dose where the animal produce normally and their offsprings show no mutagenic or teratogenic effect, that is used to estimate the safe dose for human. Okay, this is how this chronic toxicity studies they are performed. Very important thing here is throughout the lifespan it won't have any big monitoring, except they will allow to have next generation. Okay, that means male females will be kept together so that the offspring will be there, and for 3 generations the tests will be continued, so that there are no mutagenic or teratogenic effects are there. By the way, anybody has checked about I told you what is phocomolia and thalidomide earlier I told, whether anybody has checked? Fine. Okay, if not, you just try. This will help you to understand why this is done. Okay, try to search it, what is thalidomide tragedy. Okay, and try to find the phocomelia, what it is. So it will help you to understand why this test, particularly this chronic toxicity testing for animal lifespan, is prescribed. Okay, then the dose will be divided by the safety factor of 10, 100, 1000 or 10,000. It will be depending on the toxicity of the compound. This is very important. This is what we are going for, calculation of human dose from the animal dose. So what's your observation, what's your result we have obtained from the animal studies? We shall be either dividing it by 10, by 100, by 10, by 1000 or by 10,000 for understanding the human dose. Okay, and this is how we will decide, on what basis we will decide, so what kind of toxicity it has produced. Okay, this is already we have seen that cumulative response versus dose, and this is calculated accordingly. So when divided by 10 is there, when chronic or subchronic human exposure data identify an NOEL, that is NOAEL, no observed adverse effect level are available, and they are supported by the subchronic toxicity in other species. So already the drug or its container is available in market, and their human exposure data is available, and where we are having idea, that is what is the its non-observed adverse effect level, it is available, and also it is supported by chronic subchronic animal toxicity studies. Then we will divide by just 10. Now next, when we will go for division by 100, when good chronic toxicity data identifying novel are available for one or more animal species, but human data is not available, and when good chronic subchronic toxicity data is identifying a lowest human this lowest observable adverse effect level is known, so in that cases we will go. That means it is well proven in animals, but it is not in human. Then we will divide whatsoever per, why dose we have already taken as a per kg basis. Okay, so here we have it was taken as per kg basis. So on that basis, if it is per kg basis for animals, same thing, same dose divided by 100 will be for per kg basis on human beings. Now divide by 1000. When limited or incomplete chronic subchronic toxicity data is available, or when good chronic data that identify the lowest observable adverse effect level, but it is not available, that is which is very safe one, that is no observed adverse effect level for one or more animal species, then in that cases we have to divide it by 1000. Then next in that case, divided by 10,000. When subchronic study identifying a low L, that is low observable adverse effect level, but it is not giving the data or any idea about no observable adverse effect level, then we have to divide it by 10,000. Okay. Then the last slide. Yes, okay. So this is how we complete toxicity studies. These are very, very important. These toxicity studies help us in understanding, in identifying the human dosages. We have seen how these human dosages are calculated, and on that basis one can go for the calculations of dosages so that humans are not affected. Okay. Then same thing can be then extrapolated in human. Suppose we have divided by 10,000 and there is no toxicity, then gradually we can decrease the division, or gradually we can increase the dose in phase 0 or phase 1 of the clinical trial, and we can try to understand what is happening with that dose. So this is very, very important feature for the safety of the patients who are in future going to use. If you go for it, you will find very, very important cases. There are plenty of cases are there that because of some mistakes in this kind of safety studies, or again there can be variation because of the animal physiology and human physiology, because of various such factors, either errors in interpretation of this toxicology data, or error in performing the toxicological studies, or the difference in the physiology of animals and humans, and such multiple factors. It has been found that many drugs which are marketed were used, they have crossed phase 1 trial, phase 2 trial, phase 3 trial, phase 4 trial, and after few years the drug were withdrawn because they were found to have very, very severe adverse effects, resulting in death of the persons. So that means there was some mistake, and this can happen because of all this variety of reasons. So still, despite of taking so much care, there are many drugs which are withdrawn because of adverse effect. If somebody knows, I will happy to understand about it. If not, then I'll expect that you search it and find out the drug which were withdrawn from the market because of the adverse effect. Okay, they are not allowed, or they are banned drug, we can call. So they are banned because of their adverse effects. So here we finish the toxicological studies. That is a very, very important topic to understand how drugs are made available for human beings. So once all this preclinical data is compiled, then it is submitted to regulatory authority, and then only regulatory authority will permit to go for human trials. When the experts which are there in this regulatory authority, there are many experts which are sitting, like pharmacologists or doctors, they will think over it, and when they will find that yes, this data is acceptable, and on that basis the clinical trial will propose the human dose, and if this human dose calculation is appropriate on these factors, then they will allow for going for the human clinical trials, and on the basis of these human clinical trials, the clinical trials will be proceeding. By the time, if anything observed during clinical trial in any phase, particularly like adverse effects, then they will review why it has happened, and the drug will not allowed to be marketed. The drug will be removed from the trial, and drug will be, we call it as a death of the drug. The drug which was going to become, or which was going to be available in market, will be die there only. So this is how all the safety precautions are taken, and after all safety precautions, still, as I told you, there are many drugs, drugs they have to be withdrawn after few years of their availability in market, and that is called as a post-marketing surveillance. And then one more thing is there, we call it as a pharmacovigilance. So pharmacovigilance will gather the data from the market, and if the patients are having one or other kind of adverse effect that is noted, and particularly if they are causing mortality or morbidity, then this will be taken care of, and then the drugs may be banned, drug may be withdrawn from the market. So this is how all studies goes. This is how the toxicity studies, they help for making the safer drug, or allowing the safer drug for the patients. Are there any difficulties? Anyone is having any difficulty in this? So let me know if you have difficulties, please put it. And